Okay, now the quiz. So now I'm going to, in the following slides, as usually, give you some of the answers of the quiz. So here you can take the two quizzes that we're going to do today. So here I'm asking you about a definition. That is very easy. And here there are keywords that you need to know. What is the new complementary DNA strand? That is important. Our important part here is that it is synthesized continuously along the template. So if it's a new strain and it's synthesized continually, and the third thing that they told you is that it goes toward the replication fork in the mandatory 5' to 3'. So we know this. So they give you three traits that are important. That is the new complementary, that is synthesized continuously, and that is moved toward the replication fork. Just these two things tell you that this is the leading strand. So let's see. If you remember from the video lecture, here we have uh, one new strand that is the leading strand, and another strand that is the lagging strand. And remember that the DNA polymerase synthesizes the leading strand continuously, and it moves toward the replication fork. So this is the correct answer. On the other hand, the lagging strand is not continuous. If you see, it's broken, and it moves away from the replication fork. So remember, this is the replication fork and it's moving in the opposite direction. So the answer, very easy, this one, and the other answers are incorrect. If you remember, the semi-conservative model is talking about something completely different. So this is wrong. The DNA polymerase is an enzyme. The lagging strand, this is wrong. The DNA polymerase is another enzyme, and the other are just enzymes. So the only option that the only potential answer is the leading strand. Okay, now this is an enzyme, and here I'm asking you for, if you remember, the replication complex has a lot of proteins, and I'm asking you for a specific protein, a specific enzyme that is in charge of untwist the double helix. So this is fundamental, and where it does is at the replication fork. So if you remember, when you form the replication bubble, here you have the replication fork. So this is the bubble of replication, and here this is the replication fork. And this enzyme is not only untwisting the double helix and at the replication fork, but it's separating the two parental strands and making them available as a template. So they give you these four options. So it's very easy, and I just want that you think about it for a second, and I will answer that question in the next slide. So if you remember, in the video lecture I give you all these options, and here all these 7 enzymes, and here we have one that is very important. Here is the helicase, that is the one that unwinds the parental double helix in the replication fork. So it's in charge of opening. If it's like a zipper, the DNA, that enzyme is unwinding that and allowing them to, as is open, the you go from a double strand to a single strand, and now you can use the DNA polymerase to copy it. So the answer here is the helicase, telomeres is wrong, because the telomere, if you remember, this is a chromosome, so the telomere is this last part of the chromosome. Yeah, and it's a very important part because when you lose these telomeres, is those sections of the DNA. If you lose them, it has been found a relationship between when you lose them, you start getting older or you can develop cancer. It's not the only reason why people can develop cancer or grow old, but definitely telomeres are very important. So this is something completely different. Chromatin, this is not an answer. This is something completely different. And DNA polymerase is another enzyme that is in charge of synthesize the DNA. The template is the new strand. So only answer the helicase. Okay, now here I'm asking you not for an enzyme. I'm asking you for a structure of the DNA. So here they give you a clue that it looks like a bead. So that is wrapped with a string. So the DNA goes like this, and it bundled to the histones, and the string is DNA that wraps itself around the protein courtwise. So that is a very easy question. Think about it, and in the next slide, I will give you the answer. So as we said before, so it's very important that you understand what is a chromosome. Yeah, so read about that, and yeah, please go and make sure that you understand what is a chromosome, what is a histone, and here the answer will be a nucleosome. That is the basic structure unit of the DNA packing in eukaryotic. So if you remember, well, in prokaryotic you have this circular DNA. In eukaryotic you have the chromosomes look like this. So as in eukaryotic the DNA is so long, you have to organize it in a particular way. And if you see here, instead of tangled, the DNA is perfectly wrapped. So, and they use to wrap it nucleosomes that consist of DNA wound around 8 histones that are proteins. So this is a nucleosome, and if you see all these H3, H2A are histones, and if you see they give 2 turns, 1 and then 2. So 1 and 2 turns, and this is how you get this nucleosome. So definitely the only answer possible is the nucleosome, and remember it's a structure that is very important in the DNA because as in eukaryotics the DNA gets so long, instead of tangling they wrap it properly so it can replicate and you don't lose other parts. The other answers, they don't make any sense. So helicase is an enzyme, chromatin referred to the DNA. Okay, in this question, I ask you again about an enzyme, and this enzyme is in charge of catalyze the elongation of DNA from a 5' to a 3' direction. So here in this schematic, you can see the DNA polymerase 3, and what I want to show you is that it's composed of multiple proteins and multiple subunits. Here we are just going to focus on tell you that this is called the DNA polymerase 3, but if you take another course, a more advanced course, you will study this in more detail. And so this is the answer. Definitely a primase is in charge of is an enzyme in charge of set up the primer. Also, the DNA ligase is in charge of something completely different. That is, when you have two sequences of DNA, is going to put them together to link them. So this is another enzyme, but is wrong, and this one is also wrong. The topoisomerase, that one is the one that is in charge of releasing the tension here in the other side of the fork of replication, so releasing tension, so this is not. And the helicase is the one that is opening the double strand, as we said before, so all the other ones are wrong. So the only option that is possible is C. Okay, now here ask you something. I want to know your knowledge about the Okazaki fragment, and I give you 4 options. So here we have, again, if you see, you have the lagging strand, and in the lagging strand, if you remember, these sections are not synthesized continuously in the lagging strand. So in addition to replicate away from the replication fork, they have this series of fragments that are called Okazaki fragments, which are joined by a DNA ligase. So if you remember each one of this one, here you have to set up first an RNA section, and then you put the DNA besides that. So the only answer that is possible, the only one that is related to the Okazaki fragment, is the lagging strand. The premature aging disorder are actually related to the loss of telomeres, as we talked before. So there is no relation between Okazaki fragment and telomeres or premature age disorders. And the Okazaki fragments are created by restriction enzymes. This is not restriction enzymes are tools that are used in molecular biology that are like scissors. Yeah, let's draw scissors, and they are able to cut the DNA in a very specific way. So, for example, a specific restriction enzyme said I will cut if there is a sequence that is ATG, so this can allow you to cut specifically in a portion of the DNA, and in that way you can cut it and later insert another part of DNA, or you can do multiple things. So this is a very useful tool. However, in this case, it's not related to the Okazaki fragment, so again, the only option is C. Okay, now to finish this section, how do the leading and lagging strands differ from each other? So they give you these 4 options, and the right answer is A. Okay, so as we said, it was the A is the correct answer, and if you remember the schematic that I will show you in a second, we were talking that there is a lagging and a leading strand, and the leading strand is synthesized continuously, while the lagging strand you have and is going in the direction of the replication fork, while the lagging strand goes away from the replication fork and is synthesized in discontinuously. So this one is correct. A is correct. While B, if you see B says the leading strand is synthesized at twice the rate of the lagging strand. So although we know that the leading strand is synthesized faster than the lagging strand, we don't know what is the rate. So this is not true. The leading strand is synthesized by adding nucleotides to the 3' end of the growing star, and the lagging strand is synthesized by adding nucleotides in the 5'. This is wrong. Both, if you remember, both the DNA polymerase only work in one direction, so it's not that one going in the other direction and the other one going the other direction. D, the ligand strand is synthesized continuously. So this is the opposite answer of A, so this is wrong. This is discontinuously, and here the leading strand is synthesizing short fragments. So this is actually this is the definition of the ligand, and this is the definition of the leading. So this is also wrong. So the only answer is A. And here is the schematic that you have seen multiple times that show that A is the right answer.